Okay, guys, I am going to give a review of Chapter 1. So this covers a lot of material, so I'm going to do a lot of it basically as an overview, and that's kind of what I'm expecting from you guys as well. On your study guide, you have the first thing is properties of life. We'll revisit these during the course throughout in sort of many different points, really. So properties of life. That's this part here. Kind of formatting got messed up there. So, ones that we should kind of pay attention to, energy processing. So this would be like your photosynthesis and cell respiration. So things that plants do, and then cell respiration are things that all organisms do. Regulation. So this would be another word for this would be homeostasis. And a good example of that for us would be like if we start sweating when we get too hot, or if we start getting the chills, or shaking, or shivering when we get too cold. Growth and development. So this would be like in our mitosis unit. We'll talk about parts there. The thing to understand with all of these is that all of these are necessary. All are needed for something that's living. So you might have like a rock, for example. We know that rocks are not living, but they fulfill some of these requirements. So they're actually pretty highly ordered, the way that they're structured and stuff like that, but they don't reproduce. So things need to qualify under all of these conditions. Now, one of the sort of the gray areas with this is viruses. So viruses are not considered living by most biologists, even though we kind of think of them as like a germ and something that, obviously with COVID right now, spreading and all of that stuff. But viruses are not considered living because they cannot, they can't do some of these processes on their own. So when a virus infects you, it hijacks your cells and sort of takes over our cell machinery and uses that to reproduce new viruses. So viruses can do some of these processes, but they can't do all of them on their own. Viruses can do on their own without a host cell. Biological organization. That's the next section here. I'm going to start with the smallest thing because we kind of generally do that in this course. We're going to start with the smallest part. And so the smallest thing we might consider is a sugar molecule. Let me erase that because that one's not well drawn. So sugar molecules generally are hexagonal, or they can be pentagonal. So a sugar is a molecule, and you're probably most familiar if you remember earlier courses, glucose is the most common one. So when we draw these structures, you will see this is a carbon. This is a carbon, 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 carbon. And then surrounding the carbons are hydrogens. Sometimes carbons have oxygen and hydrogens attached to them. So these are all individual atoms, but this is considered a molecule, and we'll work on writing those as we go through the course. Now, we're going to kind of go upwards from here. So glucose and sugar molecules, for example, are digested in our cells, and in particular, the part of the cell that has the mitochondria. So this would be an organelle of a cell where we digest it. And that's where we get our energy from. So this part itself here is a cell, right? And inside the cell, we have things like the nucleus that houses our DNA and other cell parts as well. If we take and we make a bunch of these cells, I'm going to make them really small and tiny now, and we attach them together, then we have a part of our body called a tissue. So you might have a bunch of muscle cells, a bunch of cells in your liver or pancreas or in your brain. So that's a tissue. When you start connecting multiple tissues together, that again, these tissues are made up of cells. So tissue 1, tissue 2, tissue 3, made a bunch of cells there. So then we add all these things together and we make a organ or organ system. So the organs, what are some of the organs? Well, we have our brain, our skin, liver, pancreas, stomach. All of those are made up of different tissues, and we'll talk about some of those throughout the year. So some of these work together. So for example, your pancreas, your stomach, and your liver make up an organ system that is our digestive system. So then we go up from there. Organ system will make up an organism. So that could be a person or some sort of an animal. Let's say that we have a person and then we have a dog. And then we have a bird. When you put all of those together, these are all organisms. Then we have a community. So kind of like we have a community here in Holdingford, a community is made up of different species. Put the definition down here. Different species interacting with one another. So person and a dog can interact, dog and the bird can interact, and then you could make a lot more organisms within that community as well. Notice one of the, if you're looking at your study guide, left out the term population here. So populations are how many people do we have? How many dogs do we have? How many birds do we have? So we say population of birds, populations of people. Once we go and we take a bunch of communities and we put a bunch of communities together, then that makes up our ecosystem. And when we have a bunch of ecosystems, then that makes up what's called our biosphere. So our biosphere is all of Earth and everything living on it. Next section, next part, next couple terms is much shorter. I think the rest of the stuff is for the most part. Eukaryotic cells and prokaryotic cells. So eukaryotic cells, these are the complex ones. These make up complex organisms like people, humans, like plants, any sort of animals here, fungus. And then you have the prokaryotic cells. These are things like bacteria. One thing that's not on here is viruses again. So viruses don't classify as eukaryotic, nor do they classify as prokaryotic, but they are probably closest to bacteria. Doesn't mean that they are related or anything in that way, but they are very small in relationship to eukaryotic cells. So eukaryotic cells have all of these organelles, remember? And they have membrane, what are called membrane-bound organelles. So, for example, the nucleus in the middle here, that has a membrane around it that protects our DNA. The endoplasmic reticulum here, that has a membrane around it. Our mitochondria has a membrane around it. The Golgi body, that's this part here, that has a membrane around it. Notice none of those parts are in the prokaryotic cell. Prokaryotic cells still have DNA. And then they also have these little dots, and those are ribosomes. So those are basically the two main parts that make up a prokaryotic cell, DNA and ribosomes. So they can make protein, that's what ribosomes do, and then they have their DNA. This is a size and scale I'm going to talk about in class, but you can kind of see how big things are based off of this. So how big a person is, in the neighborhood of 1 to 2 meters in height, and then you go all the way down to a plant cell, which is somewhere between 10 and 100 micrometers, which is very small, and we need a microscope to look at those. The cutoff for our microscopes ends about right here. So we cannot see viruses with our microscope. We cannot see proteins or lipids with our microscopes. There are microscopes that exist, but not ours. DNA, genes, genomics, proteomics. So this is called the central dogma of biology. Everything that we learn about in this class is going to be drawn back to this slide. So if you don't get anything else out about this first chapter, this is something that we have to know and we're going to come back to. So we have genes, we have DNA, and those genes are housed in our nucleus. They generally send out a messenger, that is the RNA messenger. So they send out a small piece of the DNA. It gets copied, and that's what the RNA does. It copies the DNA and sends that message out, and then that makes a protein. And the proteins are the functions. Those are the things that happen in our body. So central dogma, really important. That leads to the three-domain system. Three-domain system is that we have eukaryotic cells. That's, again, the complex ones. Bacteria and archaea. So archaea is a form of bacteria, but these are the uncommon forms. We find them in really diverse locations. So they might live in a hot spring, for example. So really extreme heat, or in ice. So in really cold environments. They can live in very, very strange areas that archaea bacteria do. So we don't deal with them too much on a day-to-day basis. The bacteria that we see falls under the bacterial domain. So a domain, one of the terms on your study guide is tree of life. So if we were to draw the tree of life, and you may remember seeing this in 10th grade, you may not because you guys are distance learning, but we have the eukaryotic cells, and we are closely, not really closely related, but we are related to bacteria. And then way back here is archaea. So if we were to draw the tree of the eukaryotics, we would have, coming off of here, we would have things like plants, animals, and then fungus, all within this section of the tree. And so this just kind of shows that we are closer to a fungus and plants than we are to bacteria. Really, the only thing we have in common with bacteria is that we both have DNA and ribosomes. So kingdoms, the three-domain system is broken down into that complex kingdom, phylum, class, order, family, genus, species. The part that we most pay attention to is the genus species naming. So you would see something like Canis lupus. So canine, this comes from the Latin word. Canis means canine, and then lupus would be wolf. So versus another type would be Canis lupus familiaris. And familiaris means that we're familiar. So this is like our house dogs. So we're going to focus on the genus species naming in this course. And usually it's in 2 words there. But in reality, they actually have like 7 or 8 names because like they belong to the kingdom Animalia. So we would actually tack that on to the name of this. And they're mammals, so mammalian would be another name that we would use to tack on there. So they would get a name for each kingdom, for each phylum, each class, each order, family, genus, species. So they actually have like 7 parts to their name, but we usually just focus on the last two because we assume that we kind of know these earlier ones. Let's see, run out of room here. So this last part, last terms on things of evolution and descent with modification. So I talked earlier about DNA making RNA, making proteins, and the evolutionary part of this is how do these things change over time? And the result is a difference here, making a better protein or making a protein that's not as good. And so sometimes that happens and this becomes a fatal change or beneficial. And so the big thing here to remember is any beneficial change is the ones that are good for a species, for a population over time. And fatal ones will obviously cause them to die off. And that's descent with modification and natural selection in a nutshell. And we'll get to that mostly in second half of this course. The next part is on the scientific method. So the part to understand here, you guys did some hypothesis writing in 10th grade. Hypothesis writing, hypotheses. So if you remember the null hypothesis, the alternative 1, and the alternative 2. So this is something, 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 no change. Alternative is something, something, something increases. And alternative 2, something, something, something decreases. So if you remember those phrases, we're going to apply those when we write hypotheses. The term below there is theories, so I'm going to refer to one here in just a second. So the big thing in a theory and a hypothesis is a theory is something that takes multiple hypotheses into account and makes it something that we can understand. So for example, there is, pen keeps changing, germ theory. So germ theory is this idea that we have little organisms, and they usually have little cilia on them, so like a bacteria, for example. Or we might have something that looks more like this. And that might be a virus. Other viruses are spherical, and they have little sticky things off of them. So viruses can look like a lot of different things there. But germ theory is that these things infect us. Another theory that you learned about probably last year in chemistry was that there's atomic theory. So atomic theory is that in the middle of the atom, you have protons and you have neutrons, and these have mass. And then going around them are electrons. They orbit around, and these really don't have a mass because they're very, very light in comparison. So protons and neutrons, protons are positively charged, neutrons have zero charge, and electrons are negatively charged. So that's all part of atomic theory. Understanding the electron came from one set of hypotheses. Understanding the proton came from another set of hypotheses. And the neutron came another set of hypotheses. So, like, for example, Rutherford, the scientist named Rutherford, discovered the nucleus, or not the nucleus, but the neutron. The protons, I believe, were John Dalton. And I cannot recall the scientists who really studied electrons, but there's been lots because they have kind of the big properties. In terms of germ theory came from this outbreak of cholera in London. So people were drinking water and they were getting infected with cholera. And they used to think that germs, things that made us sick, were smelly things. But that was called the miasmatic theory. So that existed in the 1800s. I can't recall the exact year when this outbreak began, but there was a guy by the name of John Snow who started to think that that was not necessarily true, that just smelly things made us sick. And so through sort of his investigation, he found out that cholera was infecting people through the water. So it actually had to be ingested by people. So that kind of started this idea that the miasmatic theory is not correct, and that germ theory actually replaced it. So where bacteria in the water was actually making people sick. So that's theories in a nutshell. And so like he had several different hypotheses about, so hypothesis 1 plus hypothesis 2 plus hypothesis 3. He was actually working with multiple scientists, and kind of putting all those things together led this to the idea that there's these little microscopic organisms they couldn't see in the water that made them sick. So theories are made up of multiple hypotheses that are tested, and then they sort of get pieced together into a overarching concept. Germs make us sick, atoms make up all these living organisms, and so on and so forth. So then we get to inductive and deductive reasoning. So inductive and deductive reasoning are kind of challenging concepts in science. How this works, I'm going to use something that's fairly divisive in our society right now, but how we would look at something like cloth masks, for example. Since it's pretty relevant, so a couple of assumptions before we even start into this. They're porous. Viruses are smaller, from what we understand from germ theory, than those pores. And water droplets are not smaller. So, assuming that water droplets don't fit through the pores of the mask. So some fairly big assumptions there, right? I mean, water droplets can be different sizes, just like viruses can be. But if we assume that these things, then we can look at this like fairly divisive topic in two different ways. One, I'm going to start over here with the deductive side, and that is they always start with the theory. And so the theory is that germs are smaller than the pores. This is going to lead to a prediction that using masks won't help because of this overlying theory, which is correct. So yeah, these germs are smaller than the pores. So then what someone who's using deductive reasoning or a deductive investigation, they would then go into the experimental part of this, look at the data, and determine a result from that, whatever that result ends up being. The inductive reasoning part of this is that someone might make the observation that masks reduce the spread. So notice that instead of starting with a theory, they were starting with an observation on this side. So this observation might lead to viruses. So if masks are actually reducing spread, then viruses must be carried by droplets. And then this leads to what's called a paradigm. Sometimes we end up with a theory from this, but not all the time. So the paradigm here would be then that through the use of masks, it would reduce the spread of coronavirus, for example. Now, which of these is true? It doesn't really matter, right? So which of these reasoning types is true doesn't really matter. But you notice that inductive reasoning ends with the theory, or like the conclusion over the overarching concept. Whereas a deductive reasoning starts with that overarching concept and goes on to make a prediction and test that prediction. Inductive starts with the observation and ends with the understanding of the concept. So again, doesn't really matter, question mark. Like what we would have to look at is the data from this, and the data on the left part here as well. So if this is actually the case in the data, but that's how inductive and deductive reasoning are applied, which can be, like I said, a fairly challenging concept in science sometimes. So the way to think about it, deductive starts with the theory and they're testing that theory. Inductive are making an observation and sort of coming to a conclusion about that observation. I think that's about it. Oh, I got one more slide here I'm going to do. Variables, just briefly. So independent variable, IV, and dependent variable. So independent variable is the one that we change in our experimental setup. Dependent variable is the one that we're measuring as a result. So how does that change affect the measured variable that we're testing? So one example would be, how does light affect photosynthesis? So we would be measuring this in some way. We'd probably be measuring light also, but we're in control of this variable. And this one reacts in the lab. And then the last part is a control. So if we're testing the light here, we need to have controls. So controls might be the type of the plant. So if you give one plant, if you give different plants different amounts of light, different types of plants, then you're changing more than one variable. So you got to keep the type of plant the same. You've got to give them the same amount of water, and things of that nature to see how well they do photosynthesis. That's not to be confused with a controlled experiment. So in a controlled experiment, you're going to have a control group. You're going to have the testing group. So the control group would have no conditions changed. So just kind of grown naturally. So if we're doing our light experiment, that might be plants just outside, right? So then our testing group number 1, if we want them to get more light as our independent variable, increasing light, then that would be our changing condition. So more light, maybe we have them exposed to a fluorescent lamp for 24 hours, and we test to see how well they do there. On the other side of this, what we generally do is a testing group that's part of our negative testing group. So I'm going to put a negative sign here. And so a negative testing group might be that there is a decrease in light. So maybe we run the same plants under 2 hours of light, because that's less than what they would see in the outside conditions. Or maybe we do it for 0 hours and see how they react to that. So in a controlled experiment, you have a control group that really no conditions change, or we try to be as like most natural. We have a positive testing group where we would give them more light, and we have a negative testing group that would reduce the amount of light. And then we can come to a clear result. All right, that is it for chapter 1. We'll look at anything else in class.